Today we're diving into the fundamentals of heat shock transformation. This method is essential for introducing foreign DNA into bacterial cells, especially E. coli, and is widely used in genetic engineering and cloning experiments. Let's break down the process step by step. Heat shock transformation involves several key steps: preparing competent cells, mixing these cells with plasmid DNA, applying a heat shock, and finally, recovering and plating the transformed cells. First, let's talk about preparing competent cells. Competent cells are bacterial cells treated to allow them to take up DNA more easily. This is usually done by exposing the cells to a solution containing calcium chloride, which makes the cell membrane more permeable. The calcium ions neutralize the negative charges on the cell membrane, facilitating the uptake of foreign DNA. Next, we mix our competent cells with plasmid DNA. Plasmids are small circular DNA molecules that can replicate independently of the bacterial chromosome. They often carry genes of interest, such as antibiotic-resistant markers, which we can use to select for successfully transformed cells. To start, first allow the competent cells to thaw on ice for around 40 minutes before adding the plasmid. When mixing the cells, cut the end of the tip you are using to avoid damaging the cells as you pipette them up and down, or gently mix your cells by flicking the end of the tube. The mixture of competent cells and plasmid DNA is placed on ice to stabilize the cell membranes. The calcium ions neutralize the negative charges on the DNA and the cell membrane, facilitating the uptake by bringing them close to each other. Now for the heat shock step. The mixture is quickly transferred to a water bath at 42 degrees Celsius for about 45 seconds. This rapid change in temperature creates a thermal imbalance, causing the cell membranes to temporarily open, allowing the plasmid DNA to enter the cells. After the heat shock, we immediately place the cells back on ice. This helps to close the cell membrane and trap the plasmid DNA inside. The next step is recovery. We add a nutrient-rich medium like LB broth to the cells and incubate them at 37 degrees for about an hour. This allows the cells to recover from the heat shock and begin expressing the antibiotic-resistant genes carried by the plasmid. Finally, we plate the cells on agar plates containing the appropriate antibiotic. When plating, I recommend using 2 concentrations, 1/10 and 1/100, to avoid potentially crowded plates. Evenly distribute the cells and incubate overnight at 37 degrees Celsius. Only the bacteria that have taken up and expressed the plasmid DNA will grow on these plates, allowing us to select for our transformed cells. These colonies can then be further cultured or analyzed as needed for your experiment. If you found this video helpful, please give it a thumbs up and subscribe to BioLab for more tutorials and insights into molecular genetics. Leave any questions or comments below and I'll be happy to help.